Hello students, welcome to the world of recombinant DNA. In this video, we will talk about nucleases, which are called as molecular scissor. Nucleases are enzymes which are capable of cleaving the phosphodiester bond between nucleotide of nucleic acid. न्यूक्लियस बेसिकली ऐसे एंजाइम हैं जो न्यूक्लिक एसिड के स्ट्रैंड्स को ब्रेक करते हैं बाय ब्रेकिंग द फॉस्फोडाइस्टर बॉन्ड प्रेजेंट बिटवीन सक्सेसिव न्यूक्लियोटाइड्स. There are two major types of nucleases. One is exonuclease. Exo means outside. So these are the nucleases which cut the last nucleotide of a strand. They will be breaking phosphodiester bond between the last and previous nucleotide. Second type is endonuclease. These are enzymes which are capable of cutting this phosphodiester bond inside the strand of DNA. तो एक्सो मतलब बाहर, ये एंजाइम बाहर के टाइप कट करेंगे. एंडो मतलब अंदर, ये एंजाइम जो है वो स्ट्रैंड के अंदर कट करेंगे. और दिस एंजाइम्स कट एट स्पेसिफिक सीक्वेंस ऑफ अ न्यूक्लियोटाइड which is called as restriction site, which is approximately 6 to 8 base pairs. Restriction site is a site where restriction endonuclease makes the cut. So, restriction endonuclease will cut at a specific site which is called as restriction site. And generally, these restriction sites are palindrome. Now read this word. Madam, I am Adam. Now if you read this word from left to right and right to left, it will read the same. It is called as palindrome. The word that read same from forward or backward. If you read this word from left to right or right to left, the word reads same. It's called as palindrome. For example, this Hana, level, madam. If you read them backward, it will read same. Hana, level, and madam. Restriction nucleases cut DNA sequences which are palindrome. Now, genetic palindromes are similar to verbal palindromes. So, if we have a genetic sequence which read same while reading 5 to 3 prime or from 3 to 5 prime, it will read same from the both strand. For example, 5 prime G A T T C 3 prime. If you read it reverse, G 5 prime G A T T C 3 prime. They are called as genetic palindromes. Now the restriction enzymes are of 3 types. We have seen in previous video that it has both cleavage and modification site. Modification is done by methylase. So the type 1 restriction enzymes has a cleavage site which is around 1,000 base pair away from the recognition site. So recognition site will be outside the restriction site. They has endonuclease and methylase activity located on same protein molecule. तो एक ही एंजाइम कट भी करेगा और रिस्ट्रिक्शन साइट को मॉडिफाई भी करेगा. टाइप 1 का एग्जाम्पल ECOC1, ECO A, 1, CFRA1. These are examples of type 1 restriction endonucleases. Type 2, these enzyme cut a specific restriction site within the restriction site only. ये restriction site के अंदर ही कट करने वाले एंजाइम हैं टाइप 2 के. They had the endonuclease and methylase separate activities. तो एंडोन्यूक्लियस के लिए अलग से प्रोटीन का पार्ट है, मिथाइलेज के लिए अलग से प्रोटीन का पार्ट है. तो प्रोटीन कैरिंग आउट एंडोन्यूक्लियस कैन बी सेपरेटेड फ्रॉम द मिथाइलेज. फॉर एग्जांपल इको R1 है, बाम H1 है, हिन I2 है. टाइप 3 एंजाइम they cut at a random site specially located between 24 to 60 base pair away from the recognition site. तो रिकग्निशन साइट के बाहर ही कट करेंगे रैंडमली, तकरीबन 24-26 बेस पेयर दूर. एंड दे हैड endonucleases and methylase both activity located on single protein molecule. तो वही प्रोटीन मॉलिक्यूल एंडोन्यूक्लियस भी कर सकता है और मिथाइलेज मॉडिफिकेशन भी कर सकता है. इसे हम अलग से सेपरेट नहीं कर सकते. फॉर एग्जांपल है इको P1, हिम्फ I2, इको P151. तो कैन यू गेस विच type of restriction endonucleases are better for DNA technology, recombinant DNA technology? Yes, सही सोचा आपने. Type 2 restriction endonucleases can be used in the rDNA technology because they cut within the recognition site and their methylase and endonucleases activities can be separated. For example, this HAE3 is restriction endonucleases that searches the DNA molecule until it finds its 4 nitrogen base sequence, that is recognition sequence GGCEC, and it will make a cut across the GC. So HAE3 is restriction endonuclease which cut across GC bond and its recognition sequence is GGCC. Thank you.